So we'll start by talking of malaria. And as Saturday was the World Malaria Day, I guess I don't have to remind you of the enormous importance of this disease. So with over 1 million deaths per year, as you know, and over 500 million cases per year, Africa paying by far the heaviest toll with especially, unfortunately, the children under the age of 5. So the disease is due to a parasite which is called Plasmodium, which actually cycles between two hosts and schematically in 3 phases. The first phase consists in parasite replication inside red blood cells, and this causes all the symptoms and the complications of the disease. The second phase is parasite transmission by the mosquito. And the third phase is called the pre-erythrocytic phase. Basically, it extends from injection of the parasite to the first infection of red blood cells. And it mainly consists in a step of multiplication of the parasite inside hepatocyte. But other than that, so far it has been mainly a black box, which has been literally illuminated by imaging recently. And this is what I would like to convey today. Okay, so far, this pre-erythrocytic phase, at least in mammals, was considered to be a sort of a linear journey of the parasites that are deposited in the skin in small numbers, traveling through the blood and ending up in the liver, where the parasite will invade hepatocytes and progressively grow. They differentiate, they round up, they undergo nuclear divisions, and then they will bud eventually. Thousands of individual merozoites, which is the name of the stage which is adapted to infection of red blood cells. So basically, as you can understand, this step is basically a step of amplification, transformation of a few sporozoites into tens of thousands of merozoites ready to actually deliver the blood infection. Okay, so one crucial notion in the field is that attenuated sporozoites, and for a long time this has been irradiated parasites, now genetically modified parasites, are actually capable of infecting hepatocytes, but they fail to divide the nuclei, and therefore they do not produce blood infection, and then they protect their hosts. And this is true in birds, in rodents, in primates, and now in humans. It has been shown in all these recipients. And these attenuated parasites actually completely protect their hosts. This has been extensively studied in rodents, at least, with 2 main effectors of this protection: antibodies that recognize the main surface protein of the parasite and will immobilize the parasites, as well as CD8 plus T cells that will recognize parasite peptides on the surface of hepatocyte presented in conjunction with MHC class 1 molecules. So this is obviously a strong incentive to actually try to go and look further into what this pre-erythrocytic phase is about. Okay, so let me first present you the sporozoite itself. So it's a fastest, or it's one of the fastest cells. It can actually move up to 5 microns per second, and it moves by gliding. And the gliding type of movement is defined by the fact that there's no change in the shape of the cell, and these parasites actually draw circular patterns on glass slides, or I don't know if you can actually see some more corkscrew paths when they're gliding inside matrigel. So the engine of this parasite is actually a linear motor. Very simply put, it's due to, or it's powered to by myosin, which basically pulls backwards on a stable anchor, which is made of actin and a transmembrane protein. We have a number of these that directly are anchored to the substrate. So the whole phenomenon basically pulls the parasite forward. Now, this motility is obviously important for the extracellular gliding on substrate, but it can also power 2 modes of cell invasion. And that's important to know that The first one is the typical kind of cell infection. We call it cell infection, during which the parasite forms a junction, pulls on the junction, and then ends up inside a parasitophorous vacuole. You can see here the constriction of the parasite. And it's only inside this kind of parasitophorous vacuole that the parasite can actually proceed on its life cycle and start to divide. Now, a second type of cell interaction is called cell traversal, during which the parasite disrupts the plasma membrane of the host cell and will basically glide through and exit the cell. Now, you can actually image these kinds of events by incubating cells with sporozoite in the presence, for example, of cytox, which is a compound that is cell impermanent. You can place it in the extracellular medium, and it will only fluoresce when it can actually start to go inside the cell. You can see here a sporozoite that disrupts the plasma membrane and the influx of cytox inside the host cell that starts to make the host cell nucleus to fluoresce. Okay, so imaging parasites in vivo today is only possible using rodents, and that gives you the sense of the kind of very tricky system, experimental system that we have to use. So we start with mosquitoes with green fluorescent parasites here, sporozoite that are formed inside the gut of the mosquito, and then they reside in the salivary glands here. We use the two main species of Plasmodium that infect rodents, called Berghii and Uelii, and we have to allow mosquitoes to actually bite and transmit sporozoite to rodents, in this case, a hairless mouse, and then we track these extremely fast-moving objects using a high-speed spinning disk confocal microscope, which is certainly the best adapted to this kind of very motile parasites. Okay, so what we see first in the skin is that indeed most of the parasites are deposited in the skin, and they move. They move apparently randomly with some sort of a sinuous type of pattern. You can actually follow this kind of movement using maximum intensity projections, which are these red lines which basically delineates the path of the parasite during a certain amount of time on that particular cliché. It's 200 seconds, and you have a green dot, which is the sporozoite at the end of these MIPs. Now, clearly, in order to move freely in the skin, the parasites need to display this cell traversal activity that I was telling you about, because if you use the cell traversal deficient mutants, they're immediately and for good immobilized in the skin, and very rapidly those immobilized parasites are actually degraded by, in this case, resident macrophages. This time lapse is taken in a Liz GFP mouse, as well as recruited phagocytes, obviously, which are recruited very rapidly after injection of the parasite. So this cell traversal activity has more functions, but certainly a key function is to confer some resistance to the parasite to attacks by phagocytes. Okay, so by moving randomly in the skin, the parasites will eventually encounter vessels. And indeed, as expected, a number of parasites will invade blood vessels, which I show you here for one representative. Here it is. You can see the parasite here gliding in the dermis. You can see here in red and at high speed. Then it starts to go around a blood vessel, which is here in blue, makes a constriction, and then leaves with high speed with erythrocytes. So that's the typical blood vessel invasion event. Now, more unexpectedly, what we saw was that the sporozoite can also invade lymphatic vessels, which is shown in this example. Here you have a sporozoite that again glides in the dermis in red at quite high speed, then makes again this constriction, and then, unlike the case in the blood vessel, it is then drawn at very, very slow speed and in a passive and lateral way. So The sporozoite, definitely, we don't know how they do that, if it's a paracellular or a transcellular pathway, but they can invade both types of vessels. The real surprise was to notice when we did quantitative imaging, by making a lot of these studies, that only 35% of the parasites that are deposited in the skin will leave by the blood, and only 15 by the lymphatics, which means that 50% of the parasite actually stay in the skin. They're motile for a while, and then they stay once motility has disappeared. So what I'm going to do is to briefly tell you about the fate of the parasites that are gone with the blood, then the fate of those that leave with the lymph, and then we're going to come back to the skin to look at the fate of the parasite that stay in the skin. Okay, so you would think that when parasites have invaded blood vessels, they actually are passively drawn to the liver. And that's clearly not the case. I want to show you this movie. We are here in the skin, and there are lots of parasites gliding happily. Lots of very interesting things to look at in this movie. Here is an example of a sporozoite that's gliding intravascularly. But I want you to follow this one who just invaded. It's inside the vessel. The vessel corresponding is here. And then look at this one. It goes here, makes the left turn. It's really going against the blood flow. And for some time, so clearly, these parasites are very happy to glide along the endothelial cells. Now, I told you that there was a lot of things to see on that movie. So while we were following the parasite on the left, on the right, this is another very unexpected behavior. Here is a parasite that glides intravascularly, and then after a while decides to go back to the dermis and exits again by making one of these constrictions and going back to the skin. So very clearly, even the malaria parasites, I think, can be viewed as potentially some very complex and unpredictable individuals. That's really what imaging reveals us. Now, obviously, most of the parasites that are in the blood will end up in the liver, and the goal here is to invade a hepatocyte and to undergo full development and transformation, generation of merozoites. So there are quite a number of still controversial issues in the first part of this process. What I would like to show you very briefly is one very fascinating thing that we discovered with imaging, which concerns how these merozoites that are packed inside the infected hepatocyte will eventually go back to the blood in order to start the red blood cell infection cycles. So the parasites do not rupture mechanically the infected cell as was assumed, but instead they induce the formation of structures that we call merosomes. So this is a large mature parasite that contains tens of thousands of these, and you can see that the parasites are going from the infected cell into the structure that we call merosome. So this merosome contains packs of merozoites surrounded by the plasma membrane of the hepatocyte. They bud and they detach, and I'll show you on this movie the fact that they detach. So when again here, the mature parasite generates 4 merosomes, and as you can see, they stay intact in the blood flow. And a recent study by others actually showed that these merosomes are found that they can go through the right heart, and they're found in the lungs, where apparently these merosomes rupture and release the infectious merozoites. Now, these structures are not just ways to transport the parasites inside the bloodstream, but they also shield them and protect them from phagocytosis. As long as the parasites that are located inside the cell or the merosome is alive, it will pump the calcium, inhibit the activity of the scramblase enzyme, which normally positions the phosphatidylserine on the outer leaflet of the dying cell, which is normally recognized by an exin-5, and as you can see, a live parasite impedes that, and this exposure normally is recognized by macrophages. So the whole process, obviously, is a process of immune escape. The parasite remains freely unseen by the macrophages, and obviously the liver sinusoids are full of macrophages that we call Kupffer cells. Okay, now, what about the parasites that are in the lymph? We addressed that by using the footpad model, and what we found was that all the parasites that you can see here, all these little green dots, all the parasites that were inoculated are retrieved only in the proximal draining lymph node, which in the case of this experiment was the popliteal, and they don't reach the second, which in this case is the ilia. We even cannulated the thoracic duct, and we didn't see a single parasite. So they stay there, and it's clearly the end of the life cycle for those who go towards the lymphatic system. After 4 hours, about 50% of the parasites are located inside DCs. After 8 hours, we basically, most of the fluorescence has disappeared. But the interesting thing is that a few parasites can still manage to develop inside a lymph node. Now, this is the size of these rounding parasites in the node, which is not that different from the ones that you can see actually in the liver. At 24 hours, it shows clearly signs of problematic growth, if I can say so. But still, a few parasites can be seen after 24 hours. And interestingly, they're always located inside podoplaning expressing cells. And as you may know, podoplaning is a marker of endothelial cells in the lymphatic system. So in the lymph, most will be degraded, but a few will still manage to develop. Now, you remember that I told you about the protection at the beginning, which can be induced by irradiated sporozoite. Now, that's true if you inject the parasites either in the skin or IV directly. But what others have shown, the group of Fidel Zavala, is that if you immunize by injecting the parasite in the skin, then you can abrogate the protection if you remove the draining lymph node. So this is really something very important that is going on there. What we know is that the naive T cells are actually primed by activated DC inside the draining lymph node, and these will secondarily move to the liver to become the effector CD8 cells. So something important is going on in the draining lymph node. Now, back to the skin and to finish this part, we have left 50% of the parasites immotile in the skin. What do they become? Well, a proportion will again develop. I show you here 3 kinds of outcome. Here is a parasite that you follow over time, 4 days, which is slowly increasing its size. Now, this is obviously the same field. Some parasites here seem to stay approximately of the same size, and in some cases, parasite will prematurely abort. So you have all sorts of fate, but the important thing is that in the skin, the parasites can develop too, not just in the liver. In numeric terms, 10% of all the parasites that were injected actually are found developing after 24 hours. The only thing is that the growth is sort of delayed if you compare it with the growth of parasites in the liver. The maximum size will be reached at day 4, and this maximum size remains 2 to 3 times smaller than the maximum size of EF in the liver. So where do they develop? We made all sorts of cryosections that we stained with the K5 antibody, which recognized the keratin 5 inside the basal layer of keratinocytes, which delineate the dermis and the epidermis. And the response, or the answer, is that these parasites can actually develop everywhere in the skin. The majority are actually found first, in the first 2 days, in the dermis, and they seem to behave like the parasites that develop in the liver. the majority up to day 2, and then they disappear. They're also found in the epidermis, along with the layer of K5 plus keratinocytes, or in K5 negative keratinocytes, which correspond to the ones that are exfoliating in the most superficial layers. And even more surprisingly, we see a good proportion of the parasite, or actually a stable number, but a growing proportion of the parasites that associate with hair follicles that you can actually see as invaginations of the epidermis inside the dermis that are still limited by these K5 plus cells. So, and what these parasites seem to be doing, they seem to be targeting something specific close to the sebaceous glands, and we're not completely sure yet, but if we make the staining with oil red, that happens to be in green here, now you know that the sebaceous gland basically produced the sebum and lots of lipids. You can clearly see here the 2 sebaceous glands and something getting out here from the infundibulum up to the surface of the skin, and the parasite, which is in blue here, which is clearly associated. We don't know what these cells are, but there's something that the parasite really likes in this hair follicle. So in order to see that more in detail, we also used a BLIMP1 GFP mouse, in which there's a few cells which are located at or near the bud site of the sebaceous glands, emanating from the hair follicle. A few of these cells are green fluorescent, and these cells correspond to progenitor sebocytes. They give rise to the sebaceous gland. And when you infect these mice with red parasites, in this case, you see clearly that there's some sort of colocalization. So we don't know, obviously, if the parasites are located inside these stem cells or simply in the differentiated sebocyte, but there seems to be something here that the parasite truly likes. Okay, the most surprising of all was the finding that merozoites that we were convinced could only be produced in the liver can actually be produced in the skin. And we've seen that numerous times with different parasites and different mice. Using green Bergia, you can see here a schizons with numerous merozoites that are really typical of those that you can actually see in the liver. Clear schizogony. Using a red P. Bergia inside BLIMP1 GFP, you can see merozoites, red merozoites form in either BLIMP1 GFP plus or minus types of cells. And in order to test whether we were looking at something which is specific of Plasmodium berghia or something which is telling us something much more important about rodent species of malaria, we also looked at Uelii, which is supposed to more closely mimic what falciparum is doing. And in particular, both species actually invade cells in a what seems to be exquisitely dependent mechanism on CD81. And as you can see, P-Uelii2, like Berghii, actually develop inside merozoites into what seem here to be a keratinocyte in the skin. So that seems to be a general phenomenon for rodent malaria parasites. Now, with merozoites, they also produce these merosomes that I was telling you about in the liver. You can see here, after the 3rd day, this is a round EF that will all of a sudden give rise to an extension. And if you follow more in detail, with time here after 1 hour and 15 minutes, you can see that this parasite is actually giving rise to moving things. You can see here these sometimes very long extensions and clearly individual merozoites. And these things are moving in the skin. As shown here, you'll see here the EF that will give rise to actually a merosome here that moves in the skin, as weird as it may seem. Now the question was, and this is the only experiment that I'm going to show you, an obvious question was, could these skin merozoites be infectious and be responsible for red blood cell infection? And in order to test this, this is the kind of experiments that they're still ongoing. But basically, we injected in the ear of mice 30,000 sporozoite in the skin of the ear. And then we treated two groups by primaquine with quite high doses. Primaquine is known to prevent the formation of merozoites in the liver. And it happens that it does not prevent merozoite formation in the skin. We are not sure why, probably because of a lack of biodisponibility of the drug in the skin. That's our best guess, but that's what's going on. So we treat these two groups with primaquine that is supposed to actually dry up the parasites in the skin. And we also remove the ear of the third group. So we have our positive controls that are supposed to have parasites both in the skin and in the liver, our negative controls that are supposed to have parasites in neither place, and our test mice that are supposed to have parasites growing only in the skin. So how do they behave? This was our first experiment that I show you. Our positive mice are indeed positive. Our negative controls are truly negative, and we have more than 40 of these mice. They're still negative. 25 milligram per kilogram is really drying the parasite inside the liver. And then once in a while, you have these test mice that will give you an infection. So clearly, it shows that if these merozoites contribute to infection, they contribute extremely little. But I think it makes clearly the point that this development in the skin can be complete and can actually reach the stage of infectious merozoites. Okay, so I'm going to conclude here. So we have a new view of the pre-erythrocytic phase in mammalian malaria. Clearly, this phase goes from injection of the parasites to the initiation of the red blood cell cycles. We've certainly verified that they can go through via the liver to start these cycles, but this is clearly not the only fate of the parasites. Some other will go to the lymph node, and this is a dead end for the parasite life cycle. And we found that some of the parasite can actually develop, and that these can also be a source of red blood cell infecting forms. So basically we have three sites of parasite development: the skin, the lymph node, and the liver. Two sites of complete parasite development with the skin, probably most of them in the dermis, and the liver inside hepatocytes. And also an exciting new finding, which is one side of parasite persistence. We clearly still know little about that, but it's interesting because it's located along or associated with the hair follicle, which, as you know, is one of the rare sites immunoprivileged in the mammalian body. You know that there's very little MHC class 1 presentation, malfunction of antigen-presenting cells, and globally a very immunosuppressive microenvironment. So it seems to be the perfect niche for the parasite to actually grow slowly there, maybe stay dormant, and maybe at some point maybe a source of a relapse of infection. So I think also that there's one important implication. I told you about the fact that the lymph node, when parasites are delivered in the skin, is absolutely critical. What we also have learned is that this lymph node will actually see many types of antigen originating from the parasites. There's sporozoite that go directly to the lymph node, those that stay in the skin and that will come as dead parasites, or even those parasites that start to develop and that will die and secondarily will brought to this lymph node. So that clearly many questions arising, but I think the most pressing one by far, obviously you would agree with me, is how do human species behave? We have no idea, but since P. falciparum seems to frequently behave like P. euelia, and P. euelia is doing most of the things that I presented you, the question is really valid. Is there a skin phase also in human malaria? And we're going to start to image falciparum, probably both in susceptible monkeys models or maybe humanized mice, which is another possibility. Okay, I'll stop here, and I'd like to thank the people who did the work, and this is quite simple. I leave you with a parasite here. It's a sporozoite that glides for 30 minutes inside the liver parenchyma that traverses all these hepatocytes. The people that I want to thank is these people in my unit, but most importantly, Ruggero Amino, who is single-handedly did almost everything that I showed you. And two platforms at Pasteur, without which obviously this work could not have been done. The SEPIA, directed by Catherine Bourgoin, and the PFID, directed by Spencer Shorter. And I think really Ruggero and Spencer deserve your applause. Thank you very much.
Thank you, Albert. I hope you are all convinced now of the power of imaging and asking questions in this area. We have time for a few questions.
You know the proportion of the injected parasite that behavior that stay in this kind of reservoir in the pilus. So
in what kind of reservoir?
In the skin.
So in the skin. So
for the numbers that are injected.
Yeah. So I think the important thing is the general phenomenon. The proportions probably vary according to many factors. Where you inject, depending on the abdomen or the ear, for example, the degree of vascularization. In the system that we've looked carefully, which is the ear, then 50% stay, 10% are seen as round parasite at day 1, and then the proportion decreases with time, and you're going to see just a few parasites that form merozoites at day 3 or day 4. So it's very little, definitely. But you have all these antigens that are produced at different stage of development in the skin, nonetheless. So that's an important and unresolved question. We don't know the role of saliva. What we know is that the proportion of parasite that will end up in the lymph node is completely different if you inject parasites via mosquitoes or syringe. Can be more than just the absence or the presence of saliva, but clearly the mode of injection will make a whole difference in the fate of these parasites.
So when these parasites are cruising around the vessel, I'm over here.
Oh,
Hi. When they are cruising around the vessel and just when they are entering into the blood vessel, do you see any kind of vascular leakage or any signs of dysfunction of the vasculature in later stages?
No, dysfunction of the vasculature, no, because, I mean, these are extremely rare events. I remind you that we're talking about a handful of parasites that are injected in the skin in the first place. So, but it's a good question. We don't know how they actually perforate the blood vessel wall. Might be a paracellular or a transcellular pathway. We believe that it's more a paracellular pathway because the mutants that lack the capacity to traverse host cells seem to still be able to cross these endothelia. So this is something that we're starting to look at, particularly in vitro, but it seems like they're using a quite classical paracellular way to get access to the lumen.
I have a question which is not very nice, but I'll do it anyway. So you said for certain things, for example, that the parasite moves randomly in the skin. And I'm wondering if that might not be an artifact of lacking the right markers. Maybe they are following a certain pattern, I don't know, of certain molecule, of certain matrix that you're not seeing, so they're not moving randomly, and that might lead you to a wrong conclusion. Or the plasmodium that you see swimming counter current, it could be inside a cell, or there are many other explanations that might...
So the fact of moving randomly, I fully agree. what I mean by saying that they're moving randomly is that there doesn't seem to be any tactism towards the end goal and the vessels. So they move. They're probably following cues. That's obvious, because they move differently in vitro as opposed to in vivo. But they doesn't seem to exist some tactism leading them towards the blood vessel. That's what I meant.